Pleasure to introduce you to Dr. Naomi Morissette. First, I'm going to give a little brief introduction of what she's doing. Dr. Naomi is an associate professor in the Department of Molecular Biology and Biochemistry at the University of California, Irvine. She has extensive teaching experience and has presented at several conferences around the world. Additionally, she has received several grants and awards. For example, recently she received the California Research Alliance Program Award. That is actually one of the very impressive. She has mentored 17 undergrad students, 14 master students, and 8 PhD students. Throughout her career, she has published 49 articles in prestigious international journals. Furthermore, Dr. Naomi is actively involved in the collaborative research efforts, partnering with leading experts and institutions around the world. Dr. Naomi is a distinguished researcher who has made groundbreaking contributions on studying tubulin in parasites and has dedicated her career to advancing knowledge and understanding in this area. Her current research project focuses on studying tubulin and microtúbulos in apicomplex and parasites and the search of small molecules targeting tubulin. So please welcome Dr. Naomi Morissette. Thank you very much.
Thank you. Thank you for the opportunity to tell you about my work today. Today I'm going to tell you about some of the work carried out in my research laboratory that has focused on characterizing small molecules that selectively interact with protozoan tubulin. And that is because we believe that protozoan tubulin and microtubules are an overlooked and highly effective future drug target for development of therapeutic agents. Okay, so I'd like to begin by introducing you to the parasite that I work on. This is Toxoplasma. You can see that it is a unicellular organism. It's a eukaryotic organism, which of course makes design and development of drugs more complex because it shares many biochemical pathways with us, and it is a human pathogen. And so in the US, when I give this seminar, the number is about 30% of the people in this room have been infected. In Mexico, the numbers that I could find in papers vary widely. I chose the biggest one to be most spectacular, but there's a wide range of numbers. But what is important about that fact is once you're infected with Toxoplasma, you remain infected with Toxoplasma for life, okay? So I'm not a huge fan of showing lots of life cycle slides, although it gives me the opportunity to showcase one of my cats here. But I'll just briefly tell you that Toxoplasma is a parasite that infects all warm-blooded animals and can infect all nucleated cells in them. It has one particular difference when it infects cats, and that could be kitty cats or that could be lions and tigers, and that is that when cats are infected, they support the development of a sexual cycle and shed oocysts in their feces. And that allows for environmental contamination and infection of other animals. So the natural cycle should be that these oocysts are ingested by small prey animals, but they're also ingested by livestock animals, sheep, lamb, beef, and if we consume undercooked meat from these animals, we can become infected. We can also directly become infected by accidental consumption of oocysts that contaminate food or produce grown in the fields. Now, the fact that we can be infected by meat is because once this parasite completes an acute stage of infection and the immune system starts to control it, it differentiates into a slow-growing bradyzoite latent stage and forms these tissue cysts, which then are contaminating meat and are the source of the infection of the next cat or our infections. It's also possible for a woman who is pregnant and acquires an infection to pass the infection to her unborn child, or to acquire infections from transfusions or from solid organ transplants. So I'd like to just show you a little bit more about this beast before we get into talking about it, just to show you. This is a video taken by my colleague Vern Carruthers. You can see these are individual Toxoplasma. They're highly motile. We can talk later if you're interested on how they move because it's unusual, but they actively invade cells. And so this is the flat edge of a cell. This parasite has just invaded into that cell. And once it's in that cell, it is found in membrane-bound vacuoles where it replicates. And it's only in this context, within a host cell, within this parasitophorous vacuole, where replication occurs. And so parasites that are extracellular can't survive for long periods of time. So humans and other incomplete hosts support growth of this tachyzoite, or rapidly growing stage, which is what we can culture in the laboratory. Once, if tachyzoites cause the initial infection, but once immune controls set in or treatment is initiated, this slows growth of the parasite and triggers them to become the latent bradyzoite form. This is the form that persists in your body for the remainder of your life. And because these are antigenically distinct, they are in a vacuole that then has a wall around it. They are very invisible to the immune system. They're refractory to drug treatment. So neither your immune response nor any of our available drugs can eliminate these parasites. And so this becomes serious if you become immunosuppressed through any number of reasons, because the bradyzoites can redifferentiate back into tachyzoites and cause life-threatening disease. So if that weren't depressing enough, Toxoplasma is in a larger group of organisms in a phylum called the Apicomplexa, and all of these organisms are obligate intracellular parasites. So I'm sure we're all familiar with the devastation caused by Plasmodium, agent of malaria, that kills more than 500,000 people each year. Cyclospora is a second human pathogen. There's Cryptosporidium, Babesia, and Toxoplasma, which are both infections of humans and agriculturally significant livestock. And then these remaining parasites that I've circled are significant pathogens of livestock. And so these latter two categories, although we don't think of them in human health, have a large impact on economics and food production. So for all of these parasites, we have less than perfect treatment options. And one of the surprising things is that for this large array of related parasites that descended from a common ancestor, the existing treatments that we have are quite narrow spectrum. And they tend to really intersect with ways in which these parasites in different lineages have specialized. So for example, many of the drugs we use to treat malaria interfere with hemoglobin degradation, and many other apicomplexans don't invade red cells, don't use hemoglobin as a source of nutrients, and so all of these medications are ineffective against other apicomplexans. Similarly, Cryptosporidium and Cyclospora, like Toxoplasma, are orally infectious, but they remain only in the compartment of the intestines, which is a fairly hypoxic environment. And because of that, the drugs we use to treat these parasites target anaerobic metabolism. Although Toxoplasma gets in this fashion, it soon escapes and disseminates to other parts of the body, all different organ sites, including the brain. And so because of this, these drugs are not useful to eliminate or control Toxoplasma. Lastly, Toxoplasma must synthesize folates. It can't scavenge them. It doesn't have any transporters for that. And so the Drugs that we typically use as standard of care treatments inhibit folate metabolism. But some apicomplexans, notably cryptosporidium, have actually picked up the ability to transport folates and so then are insensitive to this pathway. So all of these drugs are very much narrow spectrum drugs. So an obvious choice for something that is a much broader spectrum target is tubulin and microtubules, because all eukaryotes, of course, including us, require tubulin and microtubules for cell division. So that, of course, means that if we're going to think about using tubulin as a target, we need to find ways to make compounds that are selective for inhibiting parasite tubulin. So just as a reminder, tubulin is a heterodimer of alpha and beta tubulin, and I have this video from Harvard that the first time I saw it, I cried. I thought it was so beautiful. Microtubules polymerize by stacking, subunit stacking head to tail. They depolymerize once GTP in beta tubulin is hydrolyzed, and this allows them to be extraordinarily dynamic structural components in cells, and that dynamicity is required for their function. So we have a number of agents that are either microtubule stabilizers or destabilizers, and both are toxic and interfere with processes, most notably mitosis. So we call these agents that interact with tubulin and microtubules MTAs, microtubule targeting agents, and they're used in many different contexts, including cancer chemotherapy, inflammatory disorders such as gout, for treating worm and fungal infections, and as pre-emergent herbicides. And so because tubulin has historically been such an important molecule for development of anti-cancer drugs, a lot is known about the 6 well-characterized binding pockets for small molecules that affect tubulin assembly. So of these 6 pockets that are, there are other pockets, but these are the best characterized ones, but of these 6 pockets, if you look at alignments of vertebrate tubulins and protozoan tubulins, 3 of those sites, the taxane site, the colchicine site, and the pyrinetin site, have important and conserved differences. So all vertebrates have a particular set of residues that line that pocket, and protozoa have a set of residues that line that pocket that include conserved differences. And so that leads us to believe that there are ways that we can harness these conserved differences to develop selective ligands. So the first story I'll tell you briefly about today concerns the colchicine site, which is located at the interface between beta and alpha tubulin, and ligands that bind here cause the dimer to kink so that these dimers cannot assemble into microtubules. And this work was a collaboration between my lab and the lab of Michael Steinmetz. I was at a meeting and I was sought out by Natasha and Ashwani, who are research scientists in the Steinmetz group, which is a group that's known for many co-crystal structures of small molecules and typically human tubulins for chemotherapy agents. And these guys were interested in solving the crystal structure of parasite tubulin. And initially they had this very complex plan to express parasite tubulins in a baculovirus system. And I persuaded them, based on some of our previous work that I'll tell you about later, to use tubulin from the free-living protozoan Tetrahymena thermophila as a source to crystallize as a source of protozoan tubulin. They did so. They solved the structure of the microtubule assembled from this dimer as well. And not surprisingly, it looks, at the level of the dimer, and and the microtubule quite a bit like vertebrate tubulin. But again, if you look more closely at these binding pockets, there are differences. And so specifically within the colchicine site, there are 3 positions where in vertebrate tubulins there are alanines, and within protozoan tubulins there are serines. And that suggested to Natasha and Nashwani that they could modify a colchicine site ligand to bias it to bind to protozoan tubulin. So they chose to use cumbretistatin A4, which is a natural product that binds to the colchicine site. It has the nice advantage of being a simpler natural product than some natural products. And their hypothesis was that if they extended the linker region between the A and B rings in cumbretistatin and specialize the chemistry of this linker region to optimize the polar environment of the protozoan pocket and also potentially the larger size pocket, they could have something that would be selective. And so they sent us this compound. It was mysteriously known as compound 92 when we got it, and my graduate student Isra Abali carried out assays to look at its specificity and efficacy. So like people who work on viruses, we use plaque assays to assess parasite growth, since these parasites only grow inside of host cells. So you're looking, the purple that you're seeing is staining of host fibroblast monolayers. A week earlier, they were infected with a fixed number of parasites, and these parasites invade into host cells, replicate, lyse that initial host cell, the progeny invade neighboring cells, and so you get these clear spots which represent destruction of the monolayer. And as we increase the concentration of this compound, parabolin, we see decreased size and decreased numbers of plaques, and we can use this relationship to derive an EC50 value to really have a sense of when replication is slowed by 50%. Now, of course, and I feel like this was an important control some people didn't do with screening experiments during COVID, it's important to make sure that the reason that your pathogen isn't growing is not because you've killed your host cells. And also, obviously, we want things that are selective. So what you're looking at here is a graph of values for an MTT, host cell toxicity assay, where we're comparing toxicity of cumbretistatin, which is in gray, to toxicity of parabolin. And so cumbretistatin is an investigational cancer drug. It binds to vertebrate tubulin, so not surprisingly, it has toxic effects when we treat fibroblasts, human fibroblasts with it. So over the range that we've started, it reduces the viability of host fibroblasts. In contrast, a comparable concentration of parabolin does not affect viability, although if we increase the concentration enough, we do see toxicity. But recall this EC50 value of 6.5 micromolar is going to be below this initial value here. So we have a selectivity index of about 24-fold, which is not great, but it's a reasonable starting point. So I'm going to quickly move on to the next story, and this is a story that involves a compound that we think binds to the taxane site, and that compound was first discovered by Don Wetzel, who is a physician scientist at UT Southwestern. Don and I trained together 20 years ago. We both worked on Toxoplasma together. And for those of you who are students, this is why to cherish your friendships and keep up with them, because you never know when science comes back and you meet up with people again. So it's wonderful to work with someone I've known for so long. So in the course of her research, Dawn now works on the other large group of protozoan parasites, Leishmania and trypanosomes, and she carried out a small molecule screen to look for inhibitors of Leishmania replication. And the library that she screened was a library that is available for the Medicines for Malaria venture. And so one of the compounds in this library, this one here, 477, had an effect on Leishmania that it inhibited Leishmania comparably to the standard of care treatment drugs. So that's pretty amazing as a first pass through a library. What happens is the Leishmania, I'm sorry, it's a little hard to see here, perhaps if you look at the smaller screens. In green, you're looking at tubulin, which shows you the edge of the parasites, and in blue is the DNA. And it locks parasites into these doublets where they cannot proceed further with replication. Here's a diagram from her paper that shows that you have duplicated nuclear and kinetoplastid DNA, but no division. And this suggested to her that this might be something that is preventing microtubule disassembly. And you can get a very similar phenotype if you treat Leishmania. These are extracellular cultures, so she can treat with Taxol. She doesn't have to worry about host cell effects. And so she had the sense that these were stabilizing protozoan microtubules. She did biochemistry. These compounds do indeed promote microtubule assembly selectively in protozoan parasites. We got this compound. This is Toxoplasma expressing GFP. And typically in our control cultures, there would be you would see just a sea of fluorescent parasites. They replicate so fast that they lyse all the host cells in 2 days. And in this case, we see just 2 parasites, a similar kind of locked-in phenotype that suggests that we're getting a parallel effect. So we're now working with John on derivatives of these compounds to try to improve activity. We're carrying out biochemistry. And I should say that my previous collaboration with the Steinmetz lab, we're doing the same things as well. But really, the reason I am so excited to be here is somebody that you all know really well. I am really so pleased that Rodrigo's work and my work has sort of come together. Collision isn't the right word, it's more like it's nicely intersected, and I'm really excited about how we can possibly collaborate in the future. And we are both interested in these small molecules that bind to a binding site on alpha-tubulin known as the pyrinetin site. So these small molecules, dinitroanilins, I'll talk most about oryzolin, are used commercially as herbicides. They're synthetic. They were discovered in the 1960s, and they very effectively disrupt microtubules in plant meristems, and so prevent germination. They also disrupt microtubules in protozoa, including protozoan parasites, but you can throw incredibly high concentrations of these compounds on either purified tubulin or cells from fungi or vertebrates, and there's absolutely no activity. So they are selective in the way that we're interested in. So before I show you some super old data, I will tell you that Toxoplasma has 2 primary populations of microtubules. It has spindle microtubules for cell division, and it has these cortical microtubules called subfollicular microtubules that kind of function like the ribs on your umbrella. They give the parasite its elongated and rigid shape. Oh boy, this is a little washed out, I'm sorry. In control cultures of parasites, we're using immunofluorescence staining to look at microtubule status here. In In this column, you're looking at the staining with a parasite-specific antibody for tubulin, so we don't see host cell tubulin, even though these are intracellular parasites. IMC is showing you the edge of the parasites, and DAPI, of course, is showing you DNA staining. So in the absence of treatment, you can see this parasite has replicated, and it has an elongated shape and these nice long arrays of subfollicular microtubules. If we use low levels of a dinitroaniline called ariselin, the parasites become rounded up. Maybe it's, again, look at the video monitors. They become rounded, and that's a consequence of shortened or missing subfollicular microtubules. They still have, their spindle microtubules are quite short, so they can still build spindles and undergo nuclear division. But if we increase that concentration fivefold, we completely eliminate replication of these parasites, replication being new parasites, more parasites. DNA continues to replicate, proteins continue to be synthesized. There's not the same kind of cell cycle checkpoints that other eukaryotic cells have, but the parasites are incapable of doing any cell, microtubule-based cell processes. And although both of these cultures are fine in that first host cell, once they lyse from that host cell, they cannot invade new host cells. So many years ago, I decided to select for resistance to these compounds using a chemical mutagen. I use either the low or the high concentration, and I need to remember to tell you that these parasites are haploid, so that should simplify our analysis. With both these selection conditions, we were able to isolate a large number of dependently derived lines that were resistant, and all of them, it turns out, when we analyzed them, had a mutation in alpha-tubulin. So the mutations are, this is the sequence of Toxoplasma alpha-tubulin. The mutations are in many different places in alpha-tubulin, and in some of the places where we don't have mutations in alpha-tubulin, there are plant mutations in alpha-tubulin, and in the sites where I have underlined, the same mutation has been found in both Toxoplasma and plants. We can engineer to replace the tubulin locus in wild-type parasites that are sensitive with a missense mutation from this set that I found, and show that the single missense mutation is sufficient to confer resistance. And this was done a while ago, definitely way pre-CRISPR. And so some of our transgenes went in allelically, so we had a replacement of the endogenous locus, and those gave higher resistance than when the transgene integrated elsewhere in the genome. And that makes sense, because if you have co-expression of a sensitive and a resistant tubulin, in aggregate, the tubulin population is only partially resistant. So when I started my own lab, I ended up in a biochemistry department, and they really wanted me to think about tubulin biochemistry, you know, from the point of view of true biochemistry, and not just kind of speculating based on where mutations fell, which is a bit what I had done as a postdoc. So one way that I'm sure all of you are familiar with for resistance to a drug, for a drug resistance to arise, is for a change to the binding site that blocks the ability of the drug to now bind or bind as well to that drug target. And so that's certainly something we see. But in addition, because tubulin functions as a polymer, there are mutations in both model organisms and from studies of human cancer cells that show that there are missense mutations that alter the affinity of subunits for each other. And so in the case of resistance to a destabilizing drug, you may have missense mutations that cause the dimers to make more stable microtubules. In the case of resistance to a drug like Taxol, you may have resistance mutations that cause the microtubule to be destabilized. We had some sense with dinitroanilins, our destabilizers, that resistance in some cases influence microtubule length. So these are those cortical microtubules, and these parasites, when they grow, are always the same size. Their microtubules are always the same length. And you can see that with this line here, which has been transfected with a gene with a single missense mutation, the microtubules, these subfollicular microtubules, are much longer. And so we had, this indirect sense that we were going to have stabilizing effects, at least in some cases. So in order to do biochemistry, we tried a bunch of different things. We tried budding yeast. We tried other expression systems. Tubulin is pretty finicky. It requires a eukaryotic specific set of chaperones. And so, and it also, I think it's easiest to express tubulins in something that is evolutionarily closer to the tubulin you're trying to express. So ultimately, we had success with tetrahymena, a free-living ciliate, which is kind of a fifth or sixth cousin to Toxoplasma. And it is cheap to grow, grows in media that's kind of like bacterial media. Because it's a ciliate, it has a huge amount of tubulin. That tubulin is highly conserved with Toxoplasma tubulin. It's inherently sensitive to these compounds, and it has a genetic system. So it has a single alpha tubulin gene, and just like you can do genetic swaps in yeast, we can do a similar sort of thing in tetrahymena and replace the endogenous alpha tubulin gene with an alpha tubulin gene that has a missense mutation from the set that we identified in Toxoplasma. So I'm showing you two examples of that here. And I'm showing you growth of wild-type parental tetrahymena and these two transgenic lines that only express tubulin with the missense mutation. And this is looking at growth by OD600. Wild-type tetrahymena are inhibited by, and then, slowed and then completely inhibited by growth in oryzlin. But these transgenic lines now have growth that is unimpeded by the presence of oryzlin. So that's nice, but we didn't, the reason we did this was not to do more cell biology. The reason to do this was to do biochemistry. So we can purify the tubulin from tetrahymena, and we can use it to do assays. So for example, we can use light scattering to measure tubulin assembly into microtubules. And in the presence of oryzlin, wild-type tubulin is no longer able to assemble, but this tubulin with the missense mutation from Toxoplasma is now able to assemble. We can measure binding of ligands to tubulin dimers using a technique known as tryptophan quenching. And so that allows us to derive KD values. And from these, we can see that both of the mutations that we tested in this study, both of the substitutions to the proteins reduced the affinity of oryzlin for tubulin. So we definitely get those changes to binding site affinity. In addition, you may have noticed that this assembly curve for this particular missense mutation is really quite fast. And so it reflects the fact that this particular mutation has a second effect, And that is that it also causes the tubulin dimers to have stronger affinity for each other. And we measure this by measuring something called critical concentration, which is the lowest concentration of dimers that you can have where you get microtubule assembly. So you measure this over a range of tubulin values, and the critical concentration is the intercept. And so in this particular case, wild-type tubulin and one of the two missense mutation tubulins have the same critical concentration, but this L136F tubulin has a significantly reduced critical concentrations. So that indicates that the dimers have much greater avidity for interacting. Now, people always start thinking during my talk, she says tubulin is a great drug target, but all her work is on tubulin resistance. Something's wrong here. Well, what I can tell you is that although parasites become resistant to oryzalin and other dinitroanilins, it has serious consequences for their fitness. So on this very end image, this is a normal wild-type parasite. You can see two spots of DAPI staining. This reflects the nucleus. And then these parasites have a degenerate chloroplast, and that's the chloroplast genome here. So this is the normal appearance of a healthy parasite, and all of these aberrant forms here are parasites that were created when we grew one of the resistant lines in the absence of oryzalin. And so in the absence of this drug now, they have profound defects, and not surprisingly, these guys, none of them can invade new host cells, so they're all lost to the next generation. They can't continue to invade and replicate. So resistance, although it arises, is arising in this very artificial state and is associated with considerable fitness defects. And we first appreciated this because we were worried that oryzalin might be causing mutations in the parasite genome. So we were passaging our transgenic parasites in the absence of continued selection. And so what happened is some of the resistant lines would have a big struggle replicating in this condition, but then over time they would get better. So obviously this is a sign that you may have some other genetic things happening spontaneously. And so we recloned out this line and sequenced the alpha-tubulin gene, and in two different clones we saw that the appearance of a second missense mutation. So these are most likely suppressor mutations that are compensating and helping the parasites recover from the fitness defect that is caused by the single missense mutation that confers resistance. So then what happens to resistance? Well, resistance goes down in these lines that have second site suppressors. So I'm not going to tell you this whole story, but we think this was a story about a mutation that increased microtubule stability to confer resistance, and then the suppressor mutations are actually destabilizing that increased stability. So our next question was, could we ask, is there a sweet spot? Can we get parasites that are resistant to these compounds but fit? This is important to know if you're going to make a case for tubulin being a drug target. So we took two resistant lines that had different missense mutations, and we simply passaged them for about 6 weeks in the absence of selection, and we could see that the parasites were growing increasingly more like wild-type parasites. And so ultimately we cloned out clonal populations and sequenced tubulin genes and found for both lines we had a number of second mutations in alpha-tubulin. Those lines that didn't have a second mutation in the alpha-tubulin gene had a mutation in the beta-tubulin gene for the most part. There were a few lines that we haven't followed up on that must have some other kind of mechanism to improve fitness. But so then the question was, we're improving fitness, what's the relationship with fitness and growth and resistance? And so to measure this, we again borrowed something from the yeast community, which is a competition assay. And so we have wild-type parasites that are transfected with GFP, and we mix them at a 1-to-1 ratio with each of the lines we want to analyze, and we grow them in a flask. When they lyse out, we serially pass over time. We take the remaining parasites, and we use flow cytometry to quantify the percentage of GFP parasites. And so if there's no fitness defect, in theory, we should have a 50% ratio all the way across these serial passages. The bad news that I'll tell you at the outset is our assay underestimates the fitness defects. And so this top line that you're seeing here is if we compete wild-type parasites with wild-type parasites expressing GFP. And that competition eliminates the GFP-expressing parasites in a matter of, 24 days or so. And that is most likely because expressing high expression of GFP is going to have a consequence, an extra burden for these parasites. So what that means for our assay is that it underestimates the fitness defects of our lines. So now when we do it with our lines, we're matching up the wild-type GFP parasites with the original parental strains that have just the single missense mutation in alpha-tubulin. So here's one here and one here. The colors are off, so I'm not going to refer to them by colors. I'll just say that's these two lines that are rapidly outcompeted by day 5 or 6. In contrast, if we carry out the competition assay with derivatives of these lines that now have a secondary mutation to improve fitness, you can see that while they still are outcompeted, they persist for longer. So because this was an experiment, you might imagine, done in triplicate by flow cytometry, every 2 days for a month. I couldn't ask my students to do this for more of the lines. We have nearly 50 lines. So we did a little negotiation, and we decided to pick the 2-week point. So at the 2-week point, for these particular samples, the parental lines are absent. They've been competed away, but the suppressor lines are still present at some concentration. So we felt that this was an informative window to look at. So we took the rest of those 50 lines and just serially passaged without assaying every other day, and at 14 days looked at the percentage. So this is a super confusing figure that I made myself, and so I always have to remind myself what it looks like. So the gray bars reflect resistance, so micromolar resistance to oryzolin. The black bars represent fitness, so the percentage of each of the lines in culture at 14 days. So at the very end, here's the parental line that just has the original missense mutation. So it's gone from the culture at 14 days, but it's highly resistant to oryzolin, okay? So oryzolin, look over at this axis, fitness, look over at this axis. So each of these paired sets of lines, of the black and gray lines in the bar graph, shows you the outcomes for our suppressor lines. And so although you can see that there are some lines that recover quite significant fitness, the trade-off is that they have considerably lost the ability to be resistant. Lines that retain resistance tend to have lower fitness. And so within this assay of 50 progeny, we're not identifying a sweet spot where you can have both things. So it looks like fitness and resistance are mutually exclusive. And this is something people have seen in other systems where they select for resistance in the laboratory to drugs that are used in the field. And for example, with anti-malarials, you find drugs that you can select for, you find mutations that you can select for in the laboratory that you never see in clinical populations because of these fitness costs. So when I used to give this seminar, I would end with this story about medicinal chemistry on these dinitroanilins. We tried our best to make them more drug-like. They're hydrophobic, they're nitroaromatic. Every chemist I ever talked to says what terrible compounds they are as drug leads. But with the help of Carl Werbowitz, we tried to explore this chemical space to make them better. So we did in fact find that if you added a meta-amine, we could increase efficacy of both binding to tubulin and EC50 values, but we could not get rid of these nitro groups that may make these compounds carcinogenic. And they're also, incredibly hydrophobic. So when I would give this talk, I would say these are proof of concept. They tell us that although this is not a good drug lead, it's possible for us to find ligands that are selective. And so I kind of left it at that. I was working on these other binding pockets, and Rodrigo came out with these beautiful papers that showed that the binding site was different than what we had initially thought, and that it actually is binding site that also holds ligands for vertebrate tubulin that interact with that pocket in different ways. And I am just so excited to be able to revisit looking at this binding pocket, because I think it has huge promise for us being able to design selective ligands. Okay, so this is my cue. Okay, I have a few more minutes. I often attempt to give 2-hour seminars. So I won't today, I promise. So I just wanted to tell you about some newer data. So I've told you the story of resistance to oryzolin. And so we said, piece of cake, we know how to do this. We're going to do this with parabulin and 477, those compounds I just told you about. We selected, after chemical mutagenesis, no resistant parasites. We did this several times, and then we said, okay, it's time to stop and think, why is this not working? I told you that Toxoplasma is haploid, but it has genes for 3 different alpha tubulins and 3 different beta tubulins. So there's the possibility that there's redundancy. And we hypothesize that resistance is going to be somewhat recessive to sensitivity. So 2 of the alpha tubulins are really, really strange. If you do alignments and you look at their features, they're super strange. And that leaves the remaining alpha tubulin, which is the alpha tubulin that we found the mutations in. So that's the single conventional housekeeping microtubule, alpha tubulin. The beta tubulins, all 3 are nearly identical, even at the genetic sequence, which makes them difficult to work with when you're trying to knock things out. 2 of them end up being co-expressed in the tachyzoite stage that we study. And we were able to do this by taking data that was deposited in this Parasite database and using peptides that were parallel but unique, had a unique amino acid for each of those isotypes in order to quantify the alpha and beta tubulins. So you can see Alpha-1 is nearly all the alpha-tubulin that's present. Alpha-2 and alpha-3 are negligible in terms of concentrations. 2 of the beta-tubulins are co-expressed, and the third one is just barely expressed. And so what's nice about our quantification is it nicely mirrors a genome-wide CRISPR screen done by Sebastian Laredo at MIT, which showed that, that alpha-1 followed by beta-1 and beta-2 are the most fitness-conferring, that is essential, of the tubulin genes, and the others are dispensable. So that correlates beautifully with expression. And so we thought, okay, something that's a likely Taxol site ligand, something that's a colchicine site ligand, the resistance is likely to be associated with beta-tubulin, so let's now attempt to knock out the less essential of the 2 expressed beta-tubulins. So we used CRISPR to do this, which, you know, as for many fields, has really revolutionized our lives. And so because the sequences of the tubulins were so similar, to see what we were doing, we first knocked in an in-frame YFP so that we could detect the tubulin we wanted to get rid of by green fluorescence. And, oh, I'm sorry, it's very hard to see, but there is, oh, you can see it on the other ones. Okay, so there are green fluorescent parasites, so, and with PCR we know that green fluorescence is due to an in-frame fusion of the YFP coding sequence to beta-tubulin 2. So now we take a clonal population of those green parasites, and we want to knock out that whole locus and return the parasites to not being fluorescent. And actually, I'll look here. This is what I should have been doing all along. This is just a transient snapshot. We have transiently expressed Cas9, the CRISPR machinery, and it's tagged with something that we've stained in red. And there's a parasite vacuole here, which has Cas9 staining and is no longer green. And these 2 vacuoles here have not taken up the guide RNA or Cas9, and they remain green. So when we saw this in the short-term assays, we were happy to think that we had been able to knock out the beta-2 tubulin, and ultimately we were able to use genotyping primers and show that we were able to indeed do so. Now, one of the concepts that parasitologists often use when they're looking at a potential drug target that is an essential gene is if you change the protein levels of that target, you should also then change the EC50 value. So with only one of the 2 beta-tubulins, we hypothesize that there's less tubulin dimer around, not by a huge amount, but by a significant amount, and so that we should also see a decrease then in the required concentration of drug to inhibit growth. That's what we see. We see about a quarter-fold reduction in EC50 values for our compounds. So we tried to select again. Again, we have the 2 lines now, the wild type and the beta-2 knockout, chemical mutagenesis, selection in our 3 individual drugs. And, you know, the idea is we would isolate these resistant lines. So the hypothesis for the wild type line, which has the co-expressed beta-tubulins, was that we would get dinitroaniline resistance, but not resistance to the other compounds, because we had already done this several times. That's certainly what we saw. We actually picked up more new oryzalin resistance missense mutations. But then in the beta-2 null, this is the point when your students get very sad with you. So we hypothesized that we would have mutations in beta-tubulin to confer resistance to parabolin or 477, and maybe we would even get mutations that conferred resistance to oryzalin that affected microtubule stability, but were localized to beta-tubulin. The result is we did not get resistance to parabolin or 4774 again. And curiously, we've now done this screen twice. We have 20 independently derived resistant lines in the beta knockout line. They all have one missense mutation conferring resistance to dinitroanilins. And so there's something about this missense mutation, as opposed to all the other ones we see in the diploids, that makes it much more fit or much more favorable. So why is what my students ask me, and then I have to come up with fancy answers. So what I can tell you is that parabolin and 477 don't have the same exact effects on replication. So parabolin will slow parasite replication. Many parasites will be aberrant, but it doesn't stop replication the way Arislin does. 477 at lower concentrations causes the parasites to start growing at that stage we call bradyzoites. These are latent, slow-growing forms that are pretty insensitive to all sorts of drugs. And then at the higher concentrations, where we have these locked-in doublets that I showed you, here's what's crazy. We can leave these in selection for 3 weeks, then remove the drug, and the parasites just start growing again. So they have this amazing capacity with a microtubule stabilizer to simply hunker down and stay in place. So Arislin is clearly a much more effective compound for disrupting microtubules. Selection happens more quickly, and it prevents escape by these parasites entering into latent phases. And I think that for some reason, this particular missense mutation that confers resistance is much more fit in the beta-2 null, and so that's why we have gotten it over and over again. You will recall this is the mutation that both influences Arislin binding and microtubule stability. Somehow we managed to have this be the mutation that we know about biochemically. So, almost there. Parabolin is a selective colchicine site ligand. It inhibits parasite replications. We know that microtubules are shortened and there aren't effects on vertebrate tubulin. Didn't show you that data, but we have it. We have ongoing structure function studies, which, because it's a collaboration involving intellectual property, I can't talk about. M477 stabilizes microtubules to inhibit parasite replication. Again, it's really not active on vertebrate tubulin, and we have additional structure function studies. Dinitroanilins are my favorite ligand. Alpha-1 mutations confer resistance. Missense mutations change tubulin biochemistry to reduce affinity of the tubulin dimers for dinitroanilins, to increase the dimer subunit affinity for each other. In our beta-2 null, we only found that one mutation, for some reason associated with fitness, most likely. Resistance typically reduces fitness, and there's a mutually exclusive relationship between fitness and resistance. And really the reason I'm here and I'm so excited is I'm hoping that we can design novel ligands now that because of Rodrigo's work, we understand the binding site for these molecules much better. So the people who are currently in my lab who did this work, Isra, Faliha, and Serena, they're dressed here as the Powerpuff Girls for Halloween. And my collaborators and former students, and I'm sorry, oh my God, I did go long. I'll take questions. Thank you.
Okay, thank you. Yeah. Okay, thank you very much for your talk. We have time for a couple of questions. Someone? I don't want a question. I know that the one that is introducing who's presenting has the obligation to do it, but we have been talking these days about it. So if someone have a question about it, no, actually, someone. Let's see, Jose. Don't be shy. If you want me, I can translate your question also.
Hi, thank you for your presentation. It was very interesting. Have you ever tried to assess the activity of the GTP complex in the assemble and disassemble of microtúbulos for another pathway for these experiments to try to assess the toxicity of the Toxoplasma?
So the question was, have I ever looked at, so when tubulins fold, GTP is a core ligand that's required for folding, and in beta tubulin, it's required for polymerization of microtubules. So there are really old studies that have just generally dissected that binding site, and we did try. There wasn't we've done some work on something that was alleged to be an inhibitor of the tubulin chaperonin. I'm not sure we're convinced it is, but one thing I can say, which, you know, it's funny, you can have data for years and then suddenly notice something that you never realized before. Where is the... Okay, so this this line, G142S, so for people who are tubulin aficionados, the most conserved part of tubulins is this TGS, I think is the motif, and that binds part of GTP. And that is, you know, essential for things to be considered tubulin, and it's essential for binding. And so you find it in most tubulins. I can't say all, but most tubulins. And so this is a mutation to that signature. And so maybe part of the reason this particular mutation was so deleterious was because it is influencing the ability of the dimer the monomer to fold and the dimer to assemble.
Thank you. More questions? Someone who has? Based on the GTP, I have a question. The beta tubulin has an enzymatic activity. We know the GTPase activity. So have you seen an inhibition of this GTPase activity? Or can we measure the inhibition of this? Or it would be useful at least?
Yeah. I often tell people, so I am often the crabby reviewer of papers where people express tubulins that cannot assemble because they're not in a correct expression system, but they want to claim they can measure binding of inhibitors. I would say that's like having an enzyme where you were trying to measure inhibition, but the enzyme didn't have enzymatic activity. And so I often, when I talk to people about tubulin, I try to get them to think of it as an enzyme that uses GTP hydrolysis to regulate this cycle. And so, yeah, you're right, that would be an interesting, it would have to be an in vitro assay, but is definitely informative. And there are certainly mutations that slow the GTPase rate, and those can have functional, well, there are mutations that affect things like cause spontaneous mutations that cause human developmental diseases, but then also, interestingly, some organisms have slower GTPase activity tubulins that I think are destined to be in longer-lived microtubule structures. Sorry, that was probably not clear, but
No, it's okay, thank you. Questions, more questions, someone who has? Actually, I have another question. It's just about Taxol and colchicine. Do you know if these ligands bind to the tubulin of parasites, or they don't?
So we know the efficacy of colchicine is very low for parasites. I never have measured binding, but we use the trick of getting Taxol and colchicine-resistant host cells to do these experiments. And that's often a trick we can use to kind of bypass if we want to look at a ligand that is conserved. But obviously doing the biochemical assays would be far superior.
Yeah, perfect. More questions? No? No one? Oh.
Taxol has been tested for against these parasites,
Taxol been tested.
against those parasites.
There may have been some studies in vitro, but of course, giving long-lived Taxol is, not great. So,
For human, no.
yeah.
I wonder the effect, if, just for curiosity, because you said that that was just the part of the tubulin that was tested, right?
Sorry. What I can say is my collaborator, Dawn Wetzel, her parasites can be grown outside of host cells, and those parasites, when she uses Taxol instead of 477, they have exactly the same phenotype of being locked in. So that protozoan tubulin is sensitive. Sensitive to Taxol.
Nice talk.
Thank you.
Well, ready? No more questions? Well, I want to thank again to Dr. Naomi Morissette, so please. So, we want you to give a little present from our group, and also we have this the paper that is in Spanish, but you'll see that we are going to send it the English one, so don't worry. So, gracias a todos por venir y pues esperemos ahora sí que para el siguiente seminario de la siguiente semana. Gracias.
Thank you.
Muchas gracias, doctor.
Oh, it's an axolotl.
Yeah, it's an axolotl. This is like a